Welcome to Deep Inside Biosciences. This is Graph Interpretation Part 4 for Life Sciences, Biotechnology and CSIR UGC TBT JRF Net Preparation. We have completed column and bar charts in the compare family. Now let's explore the final member of this family, the radar chart. A radar chart helps us compare many variables at the same time. Here we will compare strain A and strain B in growth, efficiency, quality, stress tolerance and yield. Let's explore how it works. Why is it called a radar chart? It is called a radar chart because its lines spread outward from one central point, like spokes on a radar screen. When values on these spokes are connected, they make a shape resembling a spider web or star. That is why it is also called a spider chart, web chart, star plot, or Kvyat diagram. But remember, a polar area chart is different and is not another name of a radar chart. Now let's understand every part of this chart step by step. First, the center is called the origin and it represents 0. Every measurement begins from this central point. Second, the lines extending outwards are called spokes. Each spoke represents one variable. Here, the 5 variables are growth, efficiency, quality, stress tolerance and yield. Third, notice the rings around the center. These rings create a scale from 0 to 5. A point closer to the center shows a lower score, whereas a point farther from the center shows a high score. Fourth, the colored dots are called data points. Each dot marks the score of a strain on one particular axis. Therefore, each variable receives its own score. Fifth, these data points are joined using connecting lines. The shape created by this line shows the overall performance profile of each strain. Sixth, always check the legend before reading the chart. Here, blue represents strain A, whereas pink represents strain B. This tells which points and lines belong to each strain. While comparing the strains, examine one trait at a time: growth, efficiency, quality, stress tolerance, and yield. On each spoke, the strain whose point lies farther from the center has the higher score. The strain whose point lies closer to the center has the lower score. Never decide by looking only at the total colored area because the area may sometimes create a misleading impression. So, the center, spokes, rings, data points, connecting lines, and legends work together to form a radar chart. Read every trait separately and the chart becomes simple to understand. Now, what does a multivariate mean? The word multivariate combines multi meaning many and variable meaning a measurable trait. Here, growth, efficiency, quality, stress tolerance, and yield are 5 different variables. Column chart compares one trait at a time, whereas a radar chart displays all these traits together. Therefore, it gives us a complete profile and helps us compare strain A and strain B across several variables in one figure. Now let us understand why must every axis in a radar chart use the same scale. Look at the chart on the left. Growth, efficiency, stress, tolerance and yield use a 0 to 5 scale, but quality uses 0 to 100. Therefore, a quality score of 80 stretches much farther than a growth score of 4. This makes quality appear unusually dominant. Even though the values use different scales, the graph therefore becomes misleading. For a fair comparison, every axis must use the same scale. If the original variables have different ranges, normalize them before plotting. Normalization means converting all values to one common scale. In the corrected chart, every trait ranges from 0 to 5, where 0 is the lowest and 5 is the highest. Now, distance from the center has the same meaning on every spoke. So, always use the same scale on every axis or normalize the values first. A common scale gives us a fair and direct visual comparison. Now let us understand in detail what does a 0 to 5 score mean. Before reading a radar chart, we must understand that a 0 to 5 score is usually not an original biological measurement. In an experiment, we may measure mass as 2.345 grams, cell concentration as 1.28 million cells per milliliter, or growth rate per hour. Because these measurements use different units, a defined conversion or normalization method changes them into one common scale from 0 to 5. For example, a growth score of 4.5 is only a relative score. It does not mean 4.5 grams, 4.5 cells, or 4.5 hours. Its meaning depends entirely on the reference used during conversions. 0 may represent none or the lowest value observed in the dataset. 5 may represent a biological maximum or simply the highest value in the dataset. This common score is useful because it allows us to compare different traits visually on one radar chart. However, we cannot look at a score of 4.5 and reconstruct the original measurement unless the conversion method is provided. Therefore, before interpreting the chart, ask 6 questions. What was originally measured? How was it converted? And what does 0 represent? What does 5 represent? Is the scale linear? Meaning, do equal steps represent equal changes? And are all traits given equal weights? So the scoring and normalization method must always be clearly defined. A 0 to 5 score has scientific meaning only when its reference and conversion method are explained. Once we know this information, we can compare the traits correctly without mistaking a relative score for an original biological unit. Now another question is, should we compare the enclosed area? The answer is no, not the area alone. Blue represents strain A and pink represents strain B. At first, a larger colored polygon may look like the better strain, but this conclusion is unreliable. These two radar charts contain exact the same values. Only the axis order has changed, yet the polygon shape and enclosed area look different. The area can also change with the scale, angles, and number of variables. Therefore, a bigger area does not mean the best overall performance. The correct method is to compare each axis. Strain A is higher in growth and stress tolerance, while strain B is higher in efficiency, quality, and yield. So use the shape only to notice the overall pattern, but use each axis for the actual comparison. Always compare trait by trait and never judge a radar chart by its enclosed area alone. Now, what do crossing lines mean? In this radar chart, blue represents strain A and pink represents strain B. Sometimes their connecting lines cross between two neighboring axes. This happens when their ranking changes. For example, on trait 1, strain A may score higher than strain B, but on the next trait, strain B may score higher than strain A. When these points are connected, the lines naturally cross. This crossing is only a geometric result of connecting the data points. It does not show a biological interaction between the traits. It also does not prove causation, correlation, or that one trait affects another. It simply shows that the strain change their relative ranking between adjacent axes. So never invent a scientific relation from crossing lines. Read the values on each scale separately, compare the strains trait by trait, and interpret only what the plotted data actually show. Now let's compare two biological strains across 5 traits. This table and radar chart compares strain A shown in blue with strain B shown in pink. Using the same 0 to 5 score scale, let us examine one trait at a time. For growth, strain A scores 4.5 while strain B scores 3.0. Therefore, strain A is higher by 1.5 score units. If we calculate the ratio, 4.5 divided by 3.0 equals 1.5. Therefore, A's growth score is 1.5 times B score or 50% higher. However, this compares normalized scores only. It does not directly prove that the actual biological growth rate is 50% higher. For efficiency, A scores 3.5 and B scores 4.2. Therefore, B is higher 0.7. For quality, A scores 4.0 and B scores 4.8, giving B an advantage of 0.8. For stress tolerance, A scores 4.5 while B scores 2.5. A is higher by 2.0, which is the largest difference in this chart. Finally, for yield, A scores 3.0 and B scores 4.0. Therefore, B is higher by 1.0. Overall, strain A performance is better in growth and stress tolerance, especially stress tolerance. Strain B performs better in efficiency, quality, and yield. Therefore, there is no single best strain for every purpose. If strong stress tolerance is our goal, strain A is the better choice. If higher yield is our goal, strain B is preferable. The correct final conclusion is that the best strain depends on the biological goal. Let's understand the trade-offs. A trade-off means that a strain may perform better in one trait but worse in another. In this radar chart, strain A shown in blue has higher growth and stress tolerance. Therefore, it is a better choice when the experimental goal is survival and continued growth under stressful conditions. Strain B shown in pink performs better in efficiency, quality and yield. Therefore, it may be better choice when the goal is high yield with good quality output. Neither strain is the strongest in all five traits. Choosing strain A gives us better growth and stress tolerance but lower efficiency, quality and yield. Choosing strain B gives us better output related traits but lower growth and stress tolerance. So a trade-off is not a mistake in the chart. It reveals different strengths and weakness. No strain is best at everything. The better choice always depends on the biological or experimental goal. If you look at the overall strain profiles, let us combine all five traits and describe each strain as a complete profile. Strain A has a growth score of 4.5, efficiency of 3.5, quality of 4.0, high stress tolerance of 4.5, and yield of 3.0. Its main strengths are strong growth and tolerance to stressful conditions such as heat, drought, and salt. It also has a good quality but comparatively low efficiency and yield. Therefore, strain A is robust, stress tolerant, and strong growing and may suit stressful conditions. Strain B has a growth score of 3.0, efficiency of 4.2, quality of 4.8, lower stress tolerance of 2.5, and yield of 4.0. Its strengths are high efficiency, very high quality, and high yield. Therefore, strain B is highest quality, high yielding option that may suit an output focused goal. So never choose a strain from one trait alone. Interpret the whole profile and select the strain that best matches the biological goal. If we want to look at which strain is the best, then we have to look at the objective because the best strain depends on the objective. Before choosing a strain, first define the biological objective. If the goal is survival under stressful conditions such as heat, drought, cold, or oxidative stress caused by ROS, strain A may be more suitable. It shows robust growth and high stress tolerance, although its yield is lower. If the goal is industrial production under stable conditions, strain B may be more suitable. It provides high efficiency, high quality, and high yield, making it useful when maximum output is important. This does not mean that one strain is universally superior. Both strains are equally valid. They are simply suitable for different purposes. Therefore, instead of asking which strain is best, ask which strain best fits the required biological objective. So, in science, best does not mean strongest in every trait. It means most suitable for the goal. Always define the objective before selecting the strain. Can we calculate an overall average score? Yes, but interpret it very carefully. For strain A, add its 5 trait scores 4.5 plus, 3.5 plus, 4.0 plus, 4.5 plus, 3.0. The total is 19.5. Dividing 19.5 by 5 gives a simple average of 3.9. For strain B, add 3.0 plus, 4.2 plus, 4.8 plus, 2.5 plus, 4.0. The total is 18.5. Dividing 18.5 by 5 gives an average of 3.7. Therefore, strain A simple average is only 0.2 higher. However, 3.9 being greater than 3.7 does not prove that strain A is biologically superior. A simple average makes 4 important assumptions. All traits are equally important. All scales have equal intervals, meaning a 1 point difference has the same meaning everywhere. The scores can be meaningfully averaged and no trait needs a special weight. Real decisions may not follow these assumptions. In industrial production, yield may matter more than other traits. In an extreme environment, stress tolerance may be essential. In such cases, a weighted average is more useful. Each trait receives a weight based on its importance, and the weighted score combines each score multiplied by its assigned weight. This allows important traits to influence the result more strongly. A simple average represents equal balance, whereas a weighted average represents goal dependent balance. Neither should replace trait by trait interpretation or the complete radar chart profile. So an average can summarize the profiles, but it cannot independently identify the best strain. The best summary and selection depends on the biological objective. Always state whether the average is simple or weighted and explain why the selected weights are appropriate. Now let us learn how to read a radar chart in 10 steps. Follow this step in order. First, read the title to understand what the chart compares. Second, check the legends to identify each color or group. Third, identify every axis and the variable shown on it. Fourth, check the scale. In this example, two strains are compared across 5 traits using a 0 to 5 scale. Fifth, remember that points near the center represents lower value, while points farther outward represents higher value. Sixth, compare the strains on one axis at a time. Seventh, find the strengths and weaknesses of each profile. Eighth, identify trade-off where a strain is better in one trait but worse in another. Ninth, never judge the enclosed area alone. 10th, match the complete profile to the biological goal. So read systematically, compare trait by trait, and interpret the chart within its biological context. Following these 10 steps help us reach a clear and careful conclusion. Now let us look where in life sciences these radar charts are found. Radar charts are useful whenever we need to compare several biological traits at once. In microbial strain screening, they can compare growth, yield, stress response, and enzyme production. For crop varieties, they can compare yield, drought tolerance, disease resistance, and quality. In phenotype profiling, they display differences among mutants, genotypes, and traits. In conservation biology, they summarize population, habitat, diversity, and traits. For drug or treatment, they can compare effects, toxicity, selectivity, and stability. They are also useful for benchmarking laboratory methods through accuracy, speed, sensitivity, and cost. Finally, bioproduct quality can be compared using purity, stability, potency, yield, and safety. In every example, we have multiple options and multiple criteria. A radar chart brings these measurements into one figure and helps us see the overall profile clearly. So, one chart can display many traits and support an informed biological selection. Now let us look into when should we use a radar chart and when we shouldn't. A radar chart is useful when we want to compare several variables at the same time and understand the overall profiles of a few groups. These variables may include performance, quality, cost, speed, and reliability. Radar chart works best with around 3 to 7 variables with only 1 or 2 variables. A simple chart is usually clearer. With 8, 9, or 10 or more variables, the chart can become crowded and difficult to read. Before plotting, every axis must use the same scale or the values must be normalized to a common scale. A radar chart is especially useful for identifying general strengths, weaknesses, and trade-off. It remains easiest to understand when only a few groups are compared. Now, when should we avoid it? Do not use a radar chart when the axes have different unnormalized scales or incompatible units. Avoid it when there are too many variables or groups because overlapping lines create confusion. It is also unsuitable when exact values are the main goal or when we need to show the data distributions and individual observations. For a precise value by value comparison, consider using a grouped bar chart, a dot plot, or a table. These formats make exact differences easier to read. A radar chart mainly answers what does the overall profile looks like. It does not clearly answer what is every exact value. So use a radar chart when 3 to 7 comparable variables, a common scale, and only a few groups. Remember, radar charts reveal overall profile, strength, and trade-off, not precise distributions. If you look at the limitations of a radar charts summarize profiles well, but they have 8 important limitations. First, exact values are difficult to read, so add data labels or a table. Second, too many variables create clutter. Third, too many groups produce overlapping lines. Fourth, a radar chart does not show the data distribution including spread, outliner, or skewness. Fifth, the position of a line does not prove statistically significance. P-values and other statistics must be shown separately. Sixth, the visual shape is sensitive to the selected scales, such as 0 to 5 versus 0 to 100. 7. Changing the axis order changes the polygon shape even when the values remain the same. 8. Normalized scores can hide original units such as centimeters, grams, or percentages. Therefore, always explain the normalization method. So, use labels, explain the scale and normalization, and report statistics separately. Remember, a radar chart summarizes overall profiles. It does not display the complete raw data. The best practices for the radar chart are: a good radar chart should be simple, clear, and scientifically honest. First, limit the number of variables, compare only a few groups, and avoid unnecessary colors. Use the same scale on every axis and clearly state whether the data are raw, normalized, or converted into scores because exact values can be difficult to read. Provide data labels or an accompanying table. Use clear axis labels, read a readable legend, and a biologically meaningful axis order. During interpretation, compare group on each axis, not by the enclosed area alone. Report the sample size, uncertainty, and present inferential statistics separately. Also explain whether a higher or lower value is biologically better for each trait. The correct workflow is to design clearly, report transparently, and interpret scientifically. So keep equal scales, clear labels, and only a few readable groups. A good radar chart makes the comparison fair, clear, and honest. If we look at the exam oriented interpretation of radar chart, in the examination mainly we'll get the questions about which is the highest growth, better quality, greatest gap, how many traits are there, and the supported conclusion. If you get this type of questions, then in an exam, read one axis at a time. Strain A leads in growth and stress tolerance. That means strain A leads 2 traits. Strain B leads in efficiency, quality, and yield. That means strain B leads 3 traits. The largest difference is in stress tolerance. 4.5 minus 2.5 equals 2.0. The growth ratio is 4.5 divided by 3.0, which equals 1.5. Therefore, A's growth score is 1.5 times B score. All axes use a common 0 to 5 scale, so trait by trait comparison is fair. If stress tolerance is the priority, strain A may suit. If yield and quality are priorities, strain B may suit. However, statistical significance cannot be inferred because the chart provides no sample size, error bars, p-values, t-test, or data distribution. The exact values may also be difficult without labels or a table. Therefore, conclude that the strains have different strengths, not that one is universally best. Read every axis, calculate carefully, and never overinterpret. If you look at the correct overall conclusion, on the common 0 to 5 scale, strain A has higher growth 4.5 compared with 3.0 and higher stress tolerance 4.5 compared with 2.5. Strain B has higher efficiency 4.2 compared with 3.5, higher quality 4.8 compared with 4.0 and higher yield 4.0 compared with 3.0. The highest difference occurs in stress tolerance 4.5 minus 2.5 equals 2.0 core units. This result shows that the strain has different strengths, not that one strain is universally better. The preferred strain therefore depends on the biological objective. Statistical significance cannot be determined from this chart alone because it provides no error bars, sample size, p-values, or statistical tests. Therefore, we cannot claim that these differences are statistically significant. So, a careful conclusion must describe the pattern, identify the differences, and respect the available evidence. Select the strain according to the biological goal and never infer significance without statistical evidence. If we look at the final takeaway of radar chart, a radar chart brings several variables together in one visual profile. Each spoke represents one variable and a point farther from the center represents higher value. However, fair interpretation requires the same scale on every axis. Read the chart trait by trait. Here, blue strain A is stronger in growth and stress tolerance, whereas pink strain B is stronger in efficiency, quality, and yield. The overall shape helps us identify strengths and trade-offs, but we should never judge the enclosed area alone. Neither profile is automatically the best. The preferred strain depends on the biological goal. We must also never claim statistical significance without supporting evidence such as sample sizes, error bars, and statistical tests. So check the axis, legends, and scale, compare every trait separately, and interpret the complete profiles within its biological context. Remember, read the axis, respect the scale, and always follow the evidence. With column chart, bar chart, and radar charts, we have completed the compare family. Next, in part 5, we will enter the change family and begin with the line graph. Until then, keep practicing. Thank you for watching this video until the end. Before we say goodbye, let us conclude with a beautiful thought. Listen to the advice and accept discipline. And at the end, you will be counted among the wise. This thought means that we should listen carefully when parents, teachers, and experienced people give us good advice. When someone corrects our mistake, we should not feel bad or become angry. We should accept the correction and improve ourselves. Listening to the advice and following discipline builds good habit, helps us learn from mistakes, and teach us to make better decisions. A person who is always willing to learn and improve gradually becomes wise. This video is also available in Telugu and Hindi. If you found it helpful, please like, share, and subscribe to Deep Inside Biosciences. Thank you.